Hey there, everyone. In the previous video, we talked a little bit about some experiments that were performed in the last 100 to 150 years that helped us to come to a modern understanding that DNA is the hereditary molecule responsible for encoding the information necessary for building proteins and basically making life possible. So, in this video, we are actually going to talk a little bit more history, but we're actually going to have kind of a lot more focus specifically on DNA structure and DNA function, and we'll talk a little bit about DNA sequencing as well. Okay, so real quick, let's review nucleic acid structure, which you'll recall is something that we discussed more thoroughly back in Chapter 3, but it may have been a while since we talked about that for you, so we'll go ahead and just review it a little bit more here. So, nucleic acids are polymers that are built by joining nucleotides together by dehydration synthesis. We do this by taking two nucleotides and we form a phosphodiester bond, that's a covalent bond, between the 3' hydroxyl of one nucleotide and the 5' phosphate of another. If you're wondering what that 3' and 5' refers to, it refers to the functional groups that are attached to different carbons that are on the pentose sugar that you see right here. In this case, with DNA, that sugar is deoxyribose. If you count the carbons, you can see from here to go clockwise, it starts 1' right there, 2' right there, 3', 4', and then 5'. You can see for this nucleotide, the alpha phosphate is attached to the 5' carbon, and the hydroxyl here that is going to be the focus of the other half of the dehydration synthesis is attached to this carbon right here. So what we're saying is that in forming a bond between two nucleotides, we are going to join the 5' phosphate of one nucleotide to the 3' hydroxyl of another nucleotide. Each nucleotide, in addition to containing a phosphate and a ribose sugar, is also going to contain a different type of nitrogenous base, and you can see the 5 different bases here. They are categorized as pyrimidines, which consist of a single cyclic ring, and then purines, which consist of 2 rings. The purines are adenine and guanine, and the pyrimidines are cytosine, thymine, and uracil. DNA only uses 4 of the 5. It uses adenine, guanine, thymine, and cytosine, and not uracil. RNA uses uracil, but not thymine. You may remember that. So in DNA, the identity of the pentose sugar is deoxyribose, which of course is missing the 2' hydroxyl that is usually found in ribose, which is found in RNA molecules. We won't get too into the weeds, but the fact that DNA is lacking that hydroxyl group actually accounts for the fact that DNA is a much more stable molecule than RNA is. And then, of course, the bases are not actually involved in forming the phosphodiester backbone of the 2 strands of DNA, so instead they just kind of hang out to the side and they're actually going to participate in complementary base pairing, which we decided in the previous video is mediated by hydrogen bonding between pyrimidines and purines. And Chargaff's rules, which tells us what bases pair with what other bases, tell us that adenine always binds with thymine or uracil in RNA, and then cytosine always bonds with guanine. And you'll notice that, again, a purine always bonds with a pyrimidine, and pyrimidines always bond with purines. You will never see pyrimidines bonding with pyrimidines and purines bonding with purines. Okay, so at this point in history, we know that DNA comes from the nuclei of cells. We know that DNA is the hereditary molecule responsible for the transformation events that were shown in Oswald Avery's experiments and then also the Hershey and Chase experiments. But we don't know what DNA looks like. We have sort of an idea of what DNA is made of, but we don't know what an intact DNA molecule actually looks like in 3-dimensional geometry. So we know at this point in current reality that DNA is maintained as a double helix with 2 complementary strands of nucleic acid winding around each other and the bases forming hydrogen bonds with one another. But think back to the 1950s. They didn't know all this. They hadn't figured all this out just yet. We can't take for granted what we know now because we are standing on the shoulders of giants. So we need to appreciate what those giants did to stick us up on their shoulders. So how was the structure of DNA solved? If we take ourselves back to the 1950s, these three gentlemen here, James Watson, Francis Crick, and Maurice Wilkins, they basically did some of the seminal work to show that DNA was indeed double helical. But we cannot, of course, forget the contributions of one Rosalind Franklin. Rosalind Franklin worked in a lab that was very focused on X-ray diffraction, and it was actually her data that directly led to the conclusion that DNA is a double helix. Now, I'm not going to pretend to be an expert in X-ray diffraction, and certainly I'm not exactly capable of interpreting X-ray diffraction data, but essentially the way this works is that if you understand that molecules are three-dimensional objects, if you bombard a molecule with X-ray radiation, then the X-rays are actually going to be bent or diffracted by the atoms that make up the molecule in question. So based on the pattern of X-rays striking a piece of film after being knocked off of atoms in a molecule, you can kind of piece together where all those atoms are attached to each other in three-dimensional space. And there was something about this X-ray diffraction pattern that you see here that led us to the conclusion that DNA must be a double helical molecule. Crick, Watson, and Maurice Wilkins, who was the graduate advisor for Rosalind Franklin, they won the Nobel Prize in Medicine in 1962, and unfortunately, you may have heard this story before, at the time, I'm not sure if this has changed, but at the time, the Nobel Prize Committee would not award Nobel Prizes posthumously, and unfortunately, Rosalind Franklin had already died by the time the award was given out, so she could not get the credit that she so richly deserved. Okay, so we can actually look at the structure of a double helix of DNA and come to the conclusion that the X-ray diffraction data are perfectly in line with kind of what we see here. We can actually see that the base pairing, as we said before, is always between a pyrimidine and a purine. So you can actually kind of come to appreciate that the purines are a little bit wider in terms of their structure because they have two rings, and the pyrimidines are a little bit more narrow. So the fact that we always, for every base pair, have a pyrimidine and a purine bonding together is in line with the X-ray data showing that DNA has a uniform diameter. If we ever were capable of having a purine-purine or pyrimidine-pyrimidine bond, that would cause the DNA molecule to be wider in some places and narrower in others. But another interesting thing that we see here, and we're not actually to the point yet where we can fully appreciate the implication of this, but we can also see that the way the DNA strands wind about each other produces both a major groove and a minor groove. So the major groove is this large opening right here, and the minor groove is this smaller opening right here. So we'll more than likely have something more to say about major and minor grooves in a later chapter, but for now, just keep in mind that certain molecules, particularly proteins, when it comes to interpreting the information that is stored in DNA, those proteins need access to the base pairs so that they can interpret the information, and it's a lot easier to access that information through the wider major groove than it is through the narrower minor groove. Okay, so given at this point that we know that DNA contains an immense amount of genetic information about how the cell builds all of its different components, namely proteins, it's actually really important from a not only scientific but technological perspective that we be able to read and interpret that information at our leisure. We ought to be able to isolate a DNA molecule and then be able to very quickly know exactly what information is contained within that molecule. And that sort of idea has been a major focus of biotechnological research and pursuits over the last few decades. You may have heard of the Human Genome Project, which was completed back in 2003, 2004, so really almost 20 years ago. But that was actually a really important undertaking because for all the genetic information that is contained in the 23 different human chromosomes, we were able to sequence all of it. We don't know exactly what it all means just yet, but we have all of the sequence of information, so we can analyze that and research it at our leisure at this point. So when we talk about DNA sequencing, it's really important that we be able to do it easily and very quickly, or at least as quickly as humanly possible. So sequencing methods did exist prior to the 1990s, but they were very slow and very laborious. So we're about to talk, on the next slide, we're about to talk about the modern method of DNA sequencing. But before we actually talk about that, we need to talk about what different components are necessary in order to synthesize a DNA molecule outside of the cell. And by outside of the cell, what I really mean is in a test tube. So to clarify here, in order to sequence a piece of DNA, you have to actually replicate the DNA, okay? So if you have a piece of DNA and you want to sequence it, you've got to make a copy of that DNA, and in making that copy, you gain the information about what sequences are actually there. So let's talk about what you actually need in order to synthesize a new DNA molecule. Now first, you have to have what is called template DNA. So in the case of sequencing, this is the source of the DNA that you want to get the information for. You want to sequence this piece of DNA. If you're not sequencing and you just want to make a copy of DNA, you've got to have some DNA to start with. You've got to have some double-stranded DNA to start with. You're going to need raw deoxynucleotides, which are sometimes called DNTPs. So these are just the building blocks that we are going to use to make our new DNA molecule. If you understand that DNA is made out of a polymer of nucleotides, then it shouldn't be a big leap in logic to say that making a new DNA molecule is going to require the building blocks. You're also going to need short oligonucleotide primers. So these are little short pieces of DNA that are complementary to the region of the DNA that you actually want to sequence. So the idea of how DNA replication and DNA sequencing is going to work in a test tube is that we are going to need to separate the two strands of DNA apart from one another. The easiest way to do that is to greatly increase the temperature. So by increasing the temperature, you break the hydrogen bonds that hold the two strands together, and you separate them into two single-stranded pieces of DNA. This, then, when you cool the temperature down, this will allow these complementary primers to bind, and then that is going to kind of serve as the starting point for new synthesis of new DNA. So from there, you can actually just attach those raw deoxynucleotides onto the end of the primer that is already bound. So in order to get new DNA to be made, you've got to at least have some double-stranded DNA to kind of tell you where to start, and then from there we can kind of take off. And then finally, there has to be an enzyme to catalyze the addition of those raw deoxynucleotides onto the end of the primer, and this enzyme is called DNA polymerase. It is literally an enzyme that polymerizes DNA molecules. So the modern method of DNA sequencing was developed by a man named Frederick Sanger. So the idea here is that he would, in a test tube, collect all four components that we just mentioned on the previous slide, except he would add a fifth component. These are called dideoxynucleotides. They're just like the regular nucleotides, except they are also missing their 3' hydroxyl in addition to their 2' hydroxyl. Now, you'll hopefully remember that 3' hydroxyl was important because that is the place where we're going to add another nucleotide onto a growing piece of DNA. So the idea here is that as the sequencing reaction is going on, as DNA polymerase is adding nucleotides, regular nucleotides, onto a growing strand of DNA, if that DNA polymerase adds a dideoxynucleotide, that terminates the chain and prevents any further nucleotides from being added because we lack that 3' hydroxyl that we need to add another base or add another nucleotide on. So once you add one of those dideoxynucleotides, you cannot extend the DNA any further. And the interesting thing is that these dideoxynucleotides were actually conjugated to fluorescent molecules, so we can detect when a particular type of dideoxynucleotide is added onto a growing chain. And by detecting this gain of fluorescence, we can actually piece together the order in which the nucleotides are found in a growing strand of DNA. And that is the essence of how DNA is sequenced. There are certainly limitations to sequencing. You can't sequence an infinitely long DNA molecule continuously. Sometimes you have to kind of do it in bits and pieces, but that is the essence and that is the basics of what you need to know for now. So another very useful application for working with DNA, not necessarily just for DNA sequencing, but agarose gel electrophoresis. So the idea here is that you make a semi-solid gel out of a sugar called agarose. We get this sugar from seaweed by mixing it together with a buffer, heating it up to dissolve the agarose in the buffer, and then allowing that liquid to cool into a semi-solid gel. You can load DNA samples into little notches that you make into the gel called wells. You can see where the DNA is being added with this multi-channel pipette here. So basically, you line all the DNA samples up at the same point, very much like how runners in a race all line up at the starting line at the same time. And then what you're going to do is you're going to run an electrical current through the gel, and this will actually cause all electrically charged molecules to migrate through the gel. Now, the nice thing about this is that all DNA molecules are negatively charged, so they will actually all be equally impacted by the electrical current, and they're all going to migrate in the same direction. But these DNA molecules will migrate at different speeds, but that speed actually has to do with how big the DNA molecule is. And we judge DNA size on the basis of how many base pairs it contains. So the idea here is that if you load your DNA samples up here, the smallest DNA molecules will reach the bottom of the gel first, and the biggest DNA molecules will kind of stay more towards the top because they are much more slowly going to move through the gel. Okay, and then this last piece of information in this video that I want to cover for now really more falls in line with some of the stuff that we talked about in Chapter 10, but I didn't really get a chance to cover this with you. It has to do with euchromatin and heterochromatin. So in previous chapters, we've obviously talked about the compaction issue of DNA and how if you stretch out DNA in a cell, it's going to be like 7 feet long. So we've talked about that and the challenge of fitting a vast amount of DNA into a very small space like the nucleus. And we talked about the role that histone proteins play in compacting the DNA. Well, the idea here is that wrapping DNA around histones kind of works one of two ways. You compact the DNA, but you also somewhat limit the access the cell has to the information that's in that DNA if you compact the DNA to too great an extent. And some regions of chromosomes actually end up more compacted than others, and this kind of causes an accessibility issue with some sequences of DNA. So this causes us to really have two different types of chromatin. Euchromatin is a much less compacted DNA that is more open and more accessible to proteins and transcription factors that are involved in making messenger RNA and synthesizing proteins. So therefore, any genes that you find in euchromatic regions can be said to be transcriptionally active. It's like a library is open for business. You can waltz in there and you can read any book that you want. You can make copies of any book that you want. So any genes that are in euchromatic regions can be very easily accessed, and we can read them and make proteins out of that information. Now, heterochromatin is the exact opposite. This is very, very densely compacted DNA that is largely inaccessible to those same transcription factors, and therefore sometimes we say that the genes here are silenced or repressed, meaning that it's like a library that is not open for business. So this difference between euchromatin and heterochromatin actually provides a very handy explanation for why different cells in the body behave differently, like heart cells behave very differently than brain cells, which behave very differently than liver cells. And we can actually explain a lot of this because certain types of cells have access to different types of information than other cells. They've all got the same information, so every cell in our body has the same 46 chromosomes, but some information that is in some cells may be more accessible than the same information in other cells. So you end up introducing a new field of study here called epigenetics, which really is the study of how gene transcription is impacted by these sorts of accessibility issues that we just mentioned. So the accessibility can be related to kind of environmental effects and behaviors rather than just the information that is contained within the DNA. So if all you're interested in is the nucleotide sequence of a DNA molecule, you're getting into the concept of genetics. But if you're interested in the sorts of factors and processes that affect how that information is accessed, then you're talking about epigenetics. All right, well, that's going to do it for this video. I certainly thank you for your attention as always. Join us next time, and we are going to start an in-depth discussion on how DNA actually gets replicated inside of cells. We sort of talked about how DNA gets replicated outside of cells, but we're really going to dive into how this happens inside of cells. Thanks again, and I will see you then. Bye-bye.